Hi, welcome to the 23rd episode of Startup Strides. Today, my guest is Gena Nikitin. Welcome, Gena.
Hi everyone. Hi David.
Gena, no, yeah. Gena is a scientist by training, and he's the co-founder of Miphic, which is a company that's working on essentially developing a platform to assess mitochondrial function and mitochondrial health and use that data for clinical applications and drug discovery. Obviously, Gena will talk more about his company in his own ways, in his own words. But before we get it started, I really want to give you a chance to kind of introduce yourself in your own words, Gena. I'm sorry I interrupted you before.
No, I just said hi everyone. No, thank you so much for the invitation. Very happy to be here. So I guess I'm Gena. I'm scientist by training. I have PhD in bioengineering, master's in organic chemistry. I also self-learned machine learning, deep learning, computer vision, Python, R, because basically that's what I apply in development of Miphic. I've been working on science, which led to Miphic for more than 12 years, and I started a company. So I broke off from academia and started an independent business 2 years ago. So here we are.
That's really cool. Well, you said you self-learned all those programming languages and machine learning. That sounds like a very, very difficult and a very lengthy process. How did you manage to do that?
No, I don't think this is that difficult. Honestly, I think that's all about the motivation and whether you need it or not. Any programming language is just a language, and in my opinion, it's not more difficult to learn programming language than any language. It's just that most of the people have no motivation to learn to program. Well, almost everyone has a motivation to communicate with others. That's how somehow people at least learn one language to communicate with the environment. And for many of the people, the ambition doesn't go further than that, and then they don't learn. Math, which is also a language. Probably my favorite language actually is math. But that's what I'm saying. It's not that difficult. And honestly, I learned it for a particular purpose because I actually learned R and Python already after I started the company, because that's what really, was a requirement to optimize the things when it started to scale on a big amount of data. Yes, and I truly believe that you cannot develop a company, especially at the early stage, if you don't know inside outs of everything which is going on. That's why I'm actively participating not only in experimental design and in the business development, but also in the code development as well.
Good.
Because
That's really cool. You said, you studied bioengineering in your PhD, and did you stay in academia before founding your company, or did you work in industry?
Yes, I did 3 years of postdoc at University of Bergen, basically yes, before starting a company here in Norway. And that was because, I was proposed to basically to have an opportunity during this postdoc to implement the technology which I was developing during my PhD, and basically got already very promising data to, for the first time ever, apply it to the clinical patient samples. And that was the very first demonstration that, yes, now it is possible to get the reliable access to mitochondrial function in the real world clinical samples. And I'm very grateful to University of Bergen for giving me this opportunity. But yes, overall, after that, definitely it was impossible to continue in academia because, well, the mission is too big to stay just, inside academia.
Got it. You already kind of touched upon that. Actually, it was my second question. I'm sorry if I'm looking to the sides because I'm trying to look at my questions. You started your startup Mythic, and it's you said it's largely based on your research and your academic research that you've done. Was it always this idea to turn this mitochondrial research into some sort of like a entrepreneurial like endeavor, or like how does that idea originate, and like what drew your attention to mitochondria in the first place?
Yeah, that was actually quite an evolution because initially, when I was 12 years ago, just in the very beginning of my PhD in Switzerland, So I actually wanted just to make something big in science, because that's why I came to science. I wanted to leave a big contribution with my life. So to basically live not for nothing. And that was my dream since I was a child. That's actually why I became a scientist to begin with. And yes, 12 years ago, basically in the beginning of my PhD, gradually identified that the fact that we ignore mitochondria in the real world is big enough of a problem to work to basically the whole life, to work on it. Because actually, why we ignore mitochondrial function, there is no good reason other than the technology did not exist that actually gives this reliable access. And yes, initially I actually spent years of not research, let's say, but working in academia, trying to implement anything that existed in terms of mitochondrial technology towards the real world applications. And I learned why we actually ignore mitochondria in the real world, because none of these technologies actually work. That's why I started to develop my own technology, which took me to develop the first, let's call it prototype, about 2.5 years, and more than 1,000 of experiments to actually get to this point, because the core enablement was through synthesizing mitochondrial pH probe, and it took me 2.5 years to synthesize the first generation of it. And that was basically my main PhD development. But then basically at the end of my PhD, I was approached by in Swiss program in Switzerland, and they evidently screen the most promising new technologies in Swiss universities. And when they see something that could be truly big, they offer you to give you a training in business and actually help you to transfer this technology to the real world. And that was how it started to evolve gradually towards the transition into the company. But yes, actually, shortly after I finished my PhD, I got this unique offer from University of Bergen to actually have a full freedom during my postdoc here to implement my technology on the clinical samples. So that was, an extremely good opportunity, and I really don't regret that I took it. So yes, that's how I moved from Switzerland to Norway. But at the same time, during my postdoc in University of Bergen, I realized that yes, there is a reason why big things, they don't stay in academia, let's say, mildly speaking. This is why it really became clear that if anything, I should have started a company earlier than just 2 years ago. But whatever, now it's developing and I'm happy with it. Yeah.
Well, I'm very glad. You started a company out of Bergen, out of Norway.
Well, yes, strictly speaking, the company is the US one. I mean, it's incorporated in Delaware, and this is where everyone is investing, who's investing in Miphic. But of course, for legal reasons, I also have a daughter company, and it is in Norway. But basically, American company fully owns the Norwegian company. So
got it. Delaware is the go-to choice for startups. And, obviously, mitochondria is very important for many reasons, but I really want to hear it from you because you've done both research and also you've worked on clinical samples, which means from the clinical side too. Can you explain why mitochondrial health is so crucial and what types of health conditions are linked to mitochondrial dysfunction, and how does Miphic aim to transform the diagnosis and treatment of these conditions?
yeah. So I guess I will give like a half a minute introduction for our viewers and listeners who don't know what mitochondria are. So mitochondria are these small things inside of our cells that actually produce all the energy we live upon. So essentially, all you need to know about mitochondria is that if they stop working, you are dead immediately. There is no life without mitochondrial function. And we actually have to breathe oxygen and eat nutrients exactly so that mitochondria burn them together to produce energy. Mitochondria do more than that, but that's the most known function of mitochondria. That said, I bet just about no one heard about mitochondria in the clinical whatever visit or something like that. I bet nobody heard that your doctor said, look, let's test your mitochondrial function. Maybe that's the reason why you are not doing so well. And so that's actually an insane situation. That was the whole point of me studying Miphic, and actually me working for more than 12 years already on that. And essentially, what we already knew extremely well through decades of preclinical research is there is no disease where mitochondrial dysfunction is not involved. So cancer is really involving the dysfunction of mitochondria, diabetes, aging, neurodegenerative diseases, and so forth. Literally, it's almost impossible to pick the disease where, in principle, you could not target mitochondria to improve the patient outcome. And everybody knows that in science and in the preclinical research. The problem is that it still stays way beyond the real world applications for the most part. And the reason is mitochondria actually start behaving poorly, not in the same way in different people, while the disease might be the same. So that if we, for example, focus on cancer, which is our key focus right now at Miphic, that's what we already know in our internal data. People with the same type of cancer, same diagnosis, same symptoms, same everything. However, what we can show, they have extremely different mitochondrial profiles. So their mitochondrial dysfunction in cancer cells is extremely different. And at the same time, these people do not respond equally to targeting of mitochondria during the pharmacological treatment. So on average, for example, in blood cancers, the best available treatment for certain types of blood cancers is a drug which targets mitochondria. However, about half of the patients do not respond to this drug. Actually, they get worse. And nobody knows why. This, again, people have same symptoms, same disease, same diagnosis. We can show now that their mitochondria work extremely differently. At the same time, we know that the drug targets mitochondria, and for some it works really well, for some it makes things worse. And that's basically, I hope, demonstrate how important it is to distinguish how exactly mitochondria work in each patient, so that we can use this information to choose the best possible treatment for each patient. That's the whole concept of precision medicine.
If you don't mind, I just want to interject a bit just to clarify for myself. You said that it's still possible to treat cancers by just targeting mitochondria, right?
Okay, so yes, if we talk about blood cancers, which I'm referring to, and this is our focus right now. I mean, of course, unfortunately, right now we cannot cure cancer. That's the unfortunate reality of the technology and medical level which we are all at yet. However, the best possible on average treatment involves targeting mitochondria. And in fact, I mean, I can mention this drug. It's called venetoclax. Venetoclax is 14th best-selling drug in the world. Not among mitochondrial drugs, but just 14th best-selling drug in the world. It has more than $2 billion of annual revenues. So that's in a way blockbuster drug, you would call. The problem is that even though that's the best drug for this type of patients, it actually doesn't help everyone. And nobody can predict whom it will help or whom it will actually make things.
Sorry, the lags there is a lag. But its mechanism of action is working on mitochondria, right?
Yes, it targets mitochondrial pathways. I mean, we can go into details, but yes, in short, it's actually prevents the ability of cells to block the ability of mitochondria to kill them. Because if our listeners or viewers are interested, basically most of healthy cells, they end their lives by suicide. Vast majority of our cells, they kill themselves once they get old, so that they don't get in the way of organism as a whole. So they're really altruistic. The only type of cells which consistently doesn't do it is cancer cells. And what also might be news for many of our listeners and viewers is that mitochondria are the key players which actually switch on the suicide mechanism. So apart from producing all of the energy, the second most important function, if not the most important maybe, function is to actually kill the cell when it's needed. When basically the cell becomes dysfunctional, it potentially starts, working against the organism and so forth. Mitochondria switch on the cellular suicide. The problem is that with in many cancer cells, this mechanism is blocked. So mitochondria develop a dysfunction when they don't kill the cell when they're supposed to anymore. And that's how cancer cells can survive, even being so dysfunctional, so not useful for the organism and so forth. And so this is the pathway that venetoclax, in short, is targeting. But it targets it without knowing what exactly happened in mitochondria. Yeah, we just do something and then we see what happens. And unfortunately, it's almost half of the patients, they don't get any better. Yeah.
The reason I was asking that question is because if the mechanism of action is based on acting on mitochondria, obviously something happening is within the mitochondria. And you said, but not everybody's responding similarly. And mitochondria is a very complex organelle, right? Because if you really even go back, there are some theories, like endosymbiotic theories, that some bacteria eventually got into this protostista and they end up turning into eukaryotic cell. So mitochondria is kind of like its own thing within the cell. It has its own DNA, it has proteins, etc., etc. When you say mitochondria are so different in their profiles, what do you mean by that? Is it the DNA that they inherit is so different, or like the proteins?
Yeah. So Miphic is primarily focusing on functional outcome. So once again, functional outcome is in our view the most important thing, because yes, of course, functional outcome also includes as upstream factors the genetic mutations, both in mitochondrial DNA and in the nuclear DNA. But the point is that genetic mutation per se is just a predisposition. It increases the risk of development of the disease or whatever, or the risk of dysfunction. The problem is that disease is not the risk. So, that's why even though people get cancer, they don't die because of the cancer the same microsecond they get it. They die when the function crosses the survival threshold. When basically cancer cells get so damaging to the whole organism, and then basically the cell, the survival of the organism cannot cope anymore with the dysfunction which is going on, then the organism actually dies. And that's why disease is actually not killing a patient immediately. And that's why, in our opinion, the most important is to target the functional outcome, because we can help a person to survive much longer, have a much higher quality of life, if we correct what dysfunctional outcome is happening inside their cells. And apart from basically this broad description of the functional approach, what is also very important that the function of mitochondria is extremely powerful in enabling cancer cells to get rid of the drugs, so be basically resistant to many drugs. That's what is also already known in the preclinical research, and that's what we're also now trying to implement in a very direct way to detect what kind of mitochondrial profile deviations are responsible for resistance to otherwise efficient therapies, so that we can, for example, block this particular dysfunction of mitochondria, so that they, again, the cancer cells are resensitized to a certain therapy to which they develop the resistance.
Oh, okay.
Because, that just maybe to, again, emphasize why it is very different and, in our opinion, really promising approach in comparison to genetic, approach. Because we actually keep getting genetic tests of patients for very, very long time. And in fact, we know that the predictive power of genetic tests is not very useful in terms of helping patients to survive. That's that's unfortunate reality, because once again, when you know that there is a genetic mutation that is responsible for a certain risk, it doesn't actually give you the chance to change this genetic mutation, right? I mean, we are not even close to the state when we easily and safely can manipulate genetics in a patient. So you see, that's very difficult to target therapeutically. However, function, for example, block some, abnormal glucose pathway or fatty acid pathway, that we actually can do. And we actually often can do it in a very efficient way. The most important is to detect what kind of functional outcome is responsible for sudden non-response to a therapy in a patient. That's our key approach, which is completely novel one as well. Yeah, in Miphic.
Got it. So just by collecting so much data, you guys are mapping different functional profiles of different clinical samples, and then you kind of guys map what functional or dysfunctional happening in mitochondria leads to the certain drug action, or like leads to not reacting to certain drug, and then you try to correct it. Is that the way, right?
Yes. So the technology essentially works the following way. We measure essentially mitochondrial battery charge, so mitochondrial pH, and then we also test how mitochondria react or don't react to different stimuli or inhibitors. For example, we can add high glucose boost, high fatty acid boost. We can add different drugs, which might be potential candidate for the patient treatment to the cells. And then we detect whether or not mitochondria react or don't react to the stimuli or inhibitions. And then we actually have a digital vector. So we have a bunch of values, which we use as the mitochondrial function vector to train the models which can predict the clinical outcomes. So this is, in summary, how it works. We have a unique access to physical workings of mitochondria, and that is mitochondrial function vector. We get the clinical outcome vector from the doctors, essentially. So which patient was, a responder or non-responder to different drugs, and we train the model which can predict based on mitochondrial profile whether or not certain drug would be efficient for this patient. That allows to choose in the future the best suited therapy for each patient. That's the concept of precision
Got it.
medicine. Yeah.
That's really cool. So how do you guys balance high sensitivity and high throughput in your mitochondrial screenings? Because I'm guessing in your work you need a lot of data, right? A lot of data. Also, you have to maintain sensitivity and be specific. So how do you balance that?
Yeah. So, we always try to acquire best possible data physically. Since Miphic is imaging-based approach, this means acquiring good quality microscopic images. And then we have proprietary image processing algorithms, which help us to reduce the noise and basically to get even higher sensitivity out of the data we get. But ultimately, of course, we also tune different parameters. So we basically do the optimizations of the tests, which I mentioned, so that, let's say, we can have the most orthogonal tests, so that basically we don't need, let's say, hundreds of tests, but it's enough, like 10 or 12 or something, which provide enough predictive power once we basically develop the models to predict the clinical outcomes. Ultimately, of course, this is the matter of basically trial and testing, and that's what we are scaling now. Our plan is to scale it to hundreds of patient samples, and then basically to validate, and that's how we're going to be getting. This is how we're going to be getting the regulatory approval to enter the market. But apart from that, Miphic is also a single cell technology. So we actually acquire one image, but this one image has hundreds of cells, right? and we actually see that different cells have different mitochondrial profiles already, and that basically helps us to really distinguish the mitochondrial dysfunctions that are responsible for ultimate response or non-response to a treatment. Because even if the cells are very different, which we already know, if we can detect all of them separately, the model starts basically, because of the granularity of the data, it starts to distinguishing between different mitochondrial profiles and whether or not they should be given weight towards the sensitivity to a certain drug. It's probably very technical if people are not familiar with machine learning and stuff, but for people who are, I'm sure they understand what I'm talking about. Yeah.
No, there are a decent number of pre-PhD or PhD people that check the episodes out, and I'm sure some people will understand. Even I have to probably go over it again just to really fully understand it. I'm curious. Well, also just to go back very quickly, we mentioned about dysfunction a lot of mitochondria, and I know there are a lot of genetical predispositions to dysfunction, the genetical risks, but at the same time, I'm curious about what external factors leads to mitochondrial function disruption later on in life. Are you aware of any particular examples?
Oh, yes, of course. That's the whole point of the functional approach, because genetics can be exactly the same, but then the real world comes with environmental factors and everything goes loose. So if we start counting the factors which are most important for mitochondria, I guess the most important are oxidative stress. If you don't eat enough antioxidants, that highly likely will increase the mitochondrial damage. If you otherwise have bad nutrition, I don't know, if you're obese, you always have a high fatty acid pressure likely on mitochondria, and they start adapting on many levels to this, and this might not be healthy for the organism as a whole. But yes, I guess one of the biggest factors on average is actually just the entropy, the universe. The universe increases entropy and things get less well functioning unless you maintain them, right? That's why it's like a toy, which or like a detail in a car. It always works, but even if you work in a completely standard conditions all the time, it gradually develops wear and tear. So unless you somehow repair it all the time, the mechanism gets older and actually gets malfunction. And so this is how I philosophically see what we're doing, at Miphic and in general, hopefully in the precision medicine approach. We are decreasing the entropy of life. We really basically helping manually to correct the basically different dysfunctions which develop because we all age ultimately, and this way we prolong the life and increase the quality of life. That's how I would say it, just decreasing the entropy of life. Yeah.
Got it. Gena, I don't want you to repeat yourself and feel like you're repeating yourself. I'm just really trying to understand, because it sounds like it's such a cool technology. Let's say you got a patient sample and you screen the mitochondria to truly understand why they're not responding to a certain drug, right? And you find the reason why. Well, you find the functional the things that went wrong, right? What's the do we have this active medicines drugs to correct most of the dysfunctionalities that you come across, or they need to be developed from now on, really?
Okay, so I would probably correct a little bit. Maybe I didn't explain it exactly the correct way before. So what I want to emphasize, the functional approach which we're using at Miphic does not necessarily find the ultimate reason why a person is a responder or non-responder to the treatment. Ultimately, we believe patient doesn't care. patient really doesn't care if we can explain to him or her, you are gonna die very soon because of these reasons, and that's exact scientific fact. Well, I don't know. And then what? Die happy or something? So what we are doing is extremely unbiased mathematical scientific approach, which is we can reliably measure mitochondrial function vector, and we're going to be developing the model which with a very high accuracy predict that if you have this mitochondrial values, let's say, in this vector, with a very high accuracy, you should be treated with certain drug because that will help you, but not with the other drug because that will actually make you worse, and you would have been better not being treated with anything rather than that drug. And that's what I'm saying. In a way, this is the model in between, right? There's a mitochondrial function vector, there's a clinical outcome vector, and then there's a model in between to which the inference is essentially being asked from. Yeah. And this is the point. In reality, probably the final models will have billions, if not trillions of parameters, maybe less, whatever. And it's very difficult to pinpoint exactly which parameter is the most crucial one. It's probably none of them will be the most crucial one. But the point is that it just works, right? You could call it even a black box. We don't mind if it will be complete black box, because as long as we can help a patient and prolong the life, whether or not we can pinpoint exactly what was that, it doesn't really matter. This is what I'm saying. This is a completely novel approach because we don't try to over-explain things. We try to be extremely practical in saying this drug would work, this drug would not.
Got it. You have a certain mitochondrial profile and you guide them to the certain treatment option based on their mitochondrial profile. Got it. Would you consider like Miphic sort of like a B2B health tech solution? Like I'm just trying to understand more about the kind of business model you guys would employ. Would it be incorporated in hospitals where like doctors could use that, or would it be essentially you guys provide this data to biotech pharma companies and force them to develop treatments? How would that work?
Well, yes, exactly. Miphic is, of course, business, B2B business. And the product is in the US, it's called Class 2 in vitro diagnostics. So essentially, that's what I just described. That's some sort of test, right? So the way basically we treat a patient sample and get our data. And it really gives you a prediction that if you have these results of the test, you should be indicated to be treated with certain drug. So class 2 in vitro diagnostics essentially being used to indicate or not indicate a certain existing drug. So it could be venetoclax, for example. But we're also, of course, targeting other metabolic drugs for now, but also we're already looking at immunotherapies as well. So this is what it is essentially doing. They're randomizing the clinical outcomes. That's our unique value for a patient, because right now, up to 60% of people who receive anti-cancer treatment, they get only worse from this treatment. no survival benefit whatsoever, only side effects. And this is why this is such a big point. That's the go-to-market strategy, to be this class 2 in vitro diagnostics, which will be able to provide the solution for essentially insurance companies to reimburse people who will be responders, and at the same time to protect patients from receiving the treatments that made them worse. Because that's an unsolved problem right now. Precision medicine is, as you probably know, the concept of the future. Unfortunately, we are not yet at the level of precision medicine in the real world clinics.
Would you like consider, for example, like big pharma or biotech firms, let's say, who are developing new mitochondrial treatments, providing data for them as a kind of like a different way of, kind of business idea for your startup?
No, sure. We always actually looking at different opportunities. And yes, these are actually, of course, potential partnerships which we are discussing now, and we get requests like that often. Yes, so I cannot actually talk publicly about any of this, but yes, things are developing in that direction as well. I can say like this: developing of the new drugs is, of course, one of the most important approaches which could be achieved with selective targeting of mitochondrial function as well, and that's what we're working with. But this is, let's say, a more classic new drug development, so therapeutic route, which is what, 10, 15 or more years before it gets to the market. Well, actually, what I talked before with this precision medicine class 2 in vitro diagnostics, this is something that we are planning to deliver to the market and to the patients, and actually start helping the patients in 2027. So this is our roadmap. In 2027, we're going to be entering the US market with this. And I actually really prefer to start helping the patients as soon as possible, especially since we have a huge problem already that patients really don't receive a treatment which they should receive, just because nobody can predict if that is the good treatment. This is why that's a cheaper problem to solve. That is a very prominent problem as it is. That's why I believe it's much more prudent from business point of view, focus on it. Well, obviously we're still working on alternatives as well. So if people want to partner with us, we are always welcome here.
Yeah, but essentially you really want to start as soon as possible and use your technology and help patients with the existing treatments that are already available in the market, right?
Yes.
Like as a world, we're really shifting, and you emphasize that several times about personalized medicine, but we're like really moving towards a more personalized medicine. And also like people are getting very interested in longevity. And obviously mitochondrial health, as you said, is one of the key aspects of it too. Do you envision a future, either with Miphic or just in general, where like people use a similar technology to individually track and optimize their mitochondrial function, and essentially like companies like yours or some other companies partnering with direct or like delivering their technology directly to the consumer, or just partnering with other wellness companies to essentially, just introduce a new longevity solution for consumers?
Yeah, I mean, absolutely. That is actually by far the most popular, external inbound requests which Miphic gets from anti-aging community and direct-to-consumer type of mitochondrial screening. That's why and it's definitely one of the most encouraging things for me, because when you talk to enthusiastic, young people who are into longevity and so forth, this is, first of all, I don't need to explain them how important mitochondria are. They actually find me to talk what we can do. Yeah. That's awesome. And second, of course, yes, that's definitely what everyone in the world will benefit at some point. Well, there is, however, a practical reason why we're still focusing on this much more regulated, basically business-to-business models like class 2 in vitro diagnostics, which require FDA approval. Because number one, the urgency of introducing this type of screening in anti-cancer treatment is profoundly, greater than in, let's say, lifestyle type of mitochondrial optimizations and so forth. So we believe that we should address the bigger problems first, than the smaller ones. And once we optimize our technology for the big problems, it will be just easier also to advertise broadly to people who are not, pioneers in longevity and so forth. And B, just from business point of view, any direct-to-consumer business is much more difficult to develop than B2B solution. Therefore, especially in the current economic climate which we're living in, I think it's better to stay in a more, basically, in a more predictable path than direct-to-consumer business. So but definitely that comes slightly later. Once we establish ourselves in the most urgent markets and regulated markets, we will definitely start developing direct-to-consumer solutions as well at some point.
Yeah, I think it's very much similar to, I was watching this talk about Neuralink and Synchron, essentially brain-computer interfaces. The goal is to start with paralyzed patients, but it will eventually move to essentially developing chips or developing this brain-computer interfaces for like general public too, but it's more down the line. I want to go back to the inception of the company and startup, and just ask you a question about the technical and business hurdles you faced along the way, like the most challenging ones, because you're a scientist by training, and academia doesn't always train you for entrepreneurship, and it's a completely different world. It's difficult, and you have to know a lot of things from IP to legal to business to fundraising to leading people. How do you navigate that?
Well, the short answer is the best way is by doing. You really cannot, read a textbook or something. It's really just better start doing it and learn from your mistakes. That's I mean, if I'm sure I'm still doing a mistake every day or several mistakes every day. The important point is just to continue going and not, because definitely if you optimize for not doing mistakes, you will never achieve anything. But I would say the biggest challenge is just that vast majority of the world doesn't know about this. Yes, we can now reliably test mitochondrial function, and that could be used to predict which therapy would be best suited for a patient. Because if people would know about this, that definitely would ease our basically scaling and entry to the market, and also it will just help so many more patients much sooner. Because yes, people are extremely conservative and mass, so my biggest challenge is often that once I show what we could do in terms of data, people are blown away. But then they're like, who are you? How come you solved this problem that nobody ever, whatever? Who are you, basically? So this is what I'm saying. Unfortunately, if you have something truly breakthrough, people get extremely skeptical about it. So on one hand, they all want something that really solves a huge problem. It really doesn't. But if it does, they always try to basically get some information by Googling, confirmation by Googling it or something. And at the moment, since we're not yet, since we didn't get the regulatory approval, this is not the case. So that's basically, I would say, the biggest challenge. To summarize, we didn't get regulatory approval yet. Thus, we are not yet recognized as something that really solves this huge problem.
Got it. What's you said it's a diagnostic, it's considered a diagnostic tool in American market by FDA, right? What are the main challenges you face in getting an approval for a product like that? What are the main kind of like the proofs that you have to show? Is it essentially what evidence do you have to really bring to move to the clinical testing stage on patients?
So you need basically to acquire enough data from clinical patient samples. I mean, the ideal situation is clinical trial data. I don't think I should be talking too much details in public in terms of what we're doing. But yes, clinical trials is part of this as well. I don't need to explain to you that clinical trials is not an easy or a cheap thing. That's why I'm not going to talk about it, but we have quite a good plan of how to make it like not many people make it, and be extremely efficient about it. But yes, it's still easier for us to enter the market because we are not developing a therapeutic. We simply
Exactly.
developing
Yeah.
a diagnostics for therapeutics which are already approved. And think about it from the patient perspective or from regulatory perspective. Once you already demonstrated, you have enough data which demonstrate that yes, if you would do this type of diagnostics, you would greatly improve the efficacy of treatment, because only people who likely respond to it will be receiving it. Who would ever want to have a Russian roulette, blind hit and miss therapy anymore?
Yeah. Yeah.
So that's why there is no market risk in that
Yeah.
sense. So yes, we really just need to scale the technology as fast as possible. That requires a lot of resources. That's and
Got
this
it.
is what we're essentially working on now.
Got it. Well, from what you said, it sounds like you guys are looking very much 0 to 1 solution, very innovative technology, and there's very much a market demand for it, right? If you really think about it, if you explain what's the issue, I feel like there is clear demand for it. Um, how's the competitive landscape like compared? Like, are there any other companies that are developing similar technology? And another thing is, I'm curious about, because you mentioned not everybody knows how important mitochondria is, or even the people that know, they don't really think about it in terms of clinical applications. So how hard was it to raise investor interest and, later on funds for your company?
Yeah, I guess I will first answer the question, which was the first one about the competitors, right?
Mm.
So, well, if we talk about direct competitors, that would be the monopolist on the market, so the Agilent Seahorse. Well, essentially, that's a company which is, I mean, everybody asks me, how is this different from Agilent Seahorse? Every new person who is our potential partner, something. So, and yes, they essentially are monopolist on the market for about a decade, since they were acquired by Agilent in 2015 for $200 million. And well, in short, the difference to them is that despite the monopoly on the market for a decade, they still have nothing going on in real world clinics. But Miphic was developed from the get-go to actually be reliable and reproducible for clinical applications. So I developed Miphic so that it can meet the problems of the existing technologies which prevented their access to real-life mitochondrial function. So I actually have zero, problems with competing. I'm actually happy to compete with them, because once people see the technological difference, it's
Yeah.
just great for me. So it's great to have such a competitor, but yeah, there is also indirect competition if we're talking about blood cancers, right? So our market entry point. This is of course the question of finding non-mitochondrial approaches to treat this cancer. Anything basically. What else could be anti-cancer treatment for these patients? And there are many approaches, but the reality is that we are so far from treating the cancer really successfully that if that gets irrelevant, I'm so happy for people. It's no problem. We're just gonna switch from blood cancers to something else. I just think this is so extremely unlikely, unfortunately, for people, that some treatment which would be found which would not require precision medicine approaches and so forth. And by the way, this is why Miphic is focusing on blood cancers. It's not that we picked it. We actually got a pull from the market. So our key core clinical partners are leukemia and other blood cancer doctors. And they actually know how important and promising targeting of mitochondria in this type of cancers. So that's why we're actually doing what they want to see in the real world clinics. That's why we don't develop something and then try to find a buyer for this or something like that. We actually directly optimizing the technology so that it can meet what they want to see in the real world. That's my approach from the beginning. I'm not just trying to push technology for someone who doesn't need it. That's why we are so much into blood cancers. That's about the competition and basically how to do it. But the second question was about fundraising, right? So the fundraising, I would say, in short, like right now, Miphic has 2 inbound investors, right? So Creator Fund and Techstars. So this year, also like more than 40 VCs contacted me, and basically I talked to them and continue talking to some of them. In short, I still did not start an active round, actually, fundraising, which I'm just gonna start soon. But yeah, I would say it's just not very productive to talk the details of fundraising in public. But yeah, I mean, I would say like that.
My question was more like, at the very beginning when you were just starting to raise funds, was it hard to generate interest in this mitochondrial technology, or were investors interested since the get-go?
Okay, so I did not start Miphic by starting raising funds. This was actually, okay, just to be clear, Miphic is working to make our customers and clinical partners happy. I really, really don't optimize to make some random investor who might invest happy. I really don't care. That's what I'm saying openly to everyone. Whoever doesn't want to be part of this, it's their loss. I have no problem with that. So that's why yes, now we are at the point when everything is ready to actually scale the technology and indeed generate enough data so that we are at a good position to start negotiating with FDA for pre-market approval of the technology, and of particular solutions for particular drugs. And this is when it becomes quite prudent to take more money from investors. That's why now I'm going to be focusing more on raising money actively. But before that, yes, Miphic got investors who were inbound and whom I liked and whom I decided to take money from, and I still like them. So yes, I really think that if your startup is optimizing to be liked by investors, you're probably not doing something big. That's one thing. Yes, and that's what I'm saying. They truly break through, revolutionary solutions, they will find investors. Again, the number one problem that not many people know that it actually exists so far, and that problem gets eventually less and less, pronounced. Also, I'm so happy to you, Javid, for this invitation, because that's part of it. Hopefully that more people know about this. Yes, and apart from that, raising money is something that is going to be happening organically once the solution is solving the real world problems. That's what I'm saying. If we really get to the point when this solves this problem that insurance companies reimburse hundreds of $1,000s only to treat every year, to treat per patient every year, to treat the consequence of unsuccessful treatment, which made the patient worse. If it truly solves it, it's impossible not to get this back. This is what I'm saying. I see that investors will compete very soon and very hard to be able to invest into Miphic. This is my view on this.
I genuinely love I admire your confidence too. It's such a good thing to really be so confident in your technology, in your science. And, but you said you've been contacted by so many VCs. And I'm curious, especially because, there are some aspiring biotech founders watching this, who are like young students, PhDs or whatever. Um, when you talk to VCs and, at the end of the day, VCs really want to, maximize the profits and revenues, right? It's the business model. So, when you talk to VCs and you have your own ideas and views, how do you how do you like choose the VCs you want to work with? Do they have to be completely fully aligned with your views and with your goal for your company, or like, what's the process, your personal selection process for working with VCs?
Yeah. So my uh, number one criterion is that yes, Miphic has a mission, which is to enable mitochondrial function in the real world. Again, one of the most basic functions of our bodies. That's why we breathe in it. This is insane that we ignore it so far. This will be completely different level of medical care once it becomes mainstream. So, number one, investors should not try to deviate from this path because they have some belief. Because this belief is almost always based on some Twitter of some person, sometimes. But yeah, because that's number one criterion. And the second criterion is that I want them to actually really help with the business development and go-to-market strategy. So since Miphic has a very defined and well discussed and well developed go-to-market strategy, such as acquire this level of data, start negotiating with FDA, get this type of clinical lab partners to distribute the diagnostics once approved and so forth. So these investors essentially should help with connections and basically introductions to the people who know very well how it's done, primarily in the US market, I guess.
Got it.
That's
That's very cool. I mean, it's been a very interesting conversation, and I really like your views and your approach to your company. And is there some founders who are like missionary founders, or there's some founders who are like called mercenary founders or opportunistic founders that just like cling on to opportunity? Yeah, but I feel like you really fit the definition of missionary founders. That you have a mission, you have a vision, you have a goal, and you're willing to do whatever it takes to get there. Which I think that's really like a very empowering energy and motivation behind a startup. We're at the end of our really episode, and feel free to obviously say whatever you want to say about Mythic and etc. I guess one final question I have for you is that, you use machine learning in your technology, obviously, and AI has been booming and being applied to literally every industry there. And it's going to be continuing to be applied and refined and increased in use. How do you envision the future of diagnostics changing with the broader application of AI and machine learning in the coming years?
Yeah. Well, AI is a tool, right?
Mm-hmm.
I mean, like many others, and that's a great tool. For Miphic, I can say that recent developments within just recent 2 years developments in computer vision really allowed me to implement my ideas and like basically single cell segmentation and so forth in such a more efficient manner, and basically to start analyzing data so much more deeply that we get with our physical screening. That yes, that was a great development. However, in my opinion, well, the biggest problem is always a problem of problems. Which problem to solve with the cutting edge tools? And that is essentially always going to be a problem for certain, hopefully smart human beings. The problem of problems, because if you think about it, the emergence of personal computers everywhere probably had more of an impact in terms of what we could do with the technology and also medicine and so forth in the world. the point is that yes, so AI, it's great. Humanity is developing. So what we call now AI, which is to me just another math development, so whatever, is great. So whoever is not adapting in using it in their basically work and applications will lose the competition. So that's why obviously we're using for particular purposes what we need. And that just basically means computer vision at Miphic. It very actively. But that said, ultimately it will be the problem of problems. So once again, I don't think patients matter. It matters for patients that we use now computer vision, whatever AI, to increase the accuracy of the prediction of our screening. For patients, definitely what matters whether or not the drug saves them or not, right? They really don't care if AI or not AI. So this is what I'm saying. But yes, AI is a great mathematical, advancement. We should continue in that direction more. Ultimately, human beings are horrible in just about everything, but their role is to choose the problems to be solved. So the problem of problems. And that's how I want to also to continue.
And using diagnostics is ripe for that, like in terms of problems that can be solved with AI.
Well, I think this is an extremely generalistic to just say diagnostics is a good application for AI, because even at Miphic, it's I actually hate to say that we're using AI. I'm just saying we're using computer vision or whatever, machine learning, and all of that is just implemented again to solve a problem of predicting the best possible treatment for a patient, because it's the best tool. I'm focusing on developing these tools. If tomorrow we will invent magic and it will work better, believe me, I will use
You're
magic
changing.
in Miphic. So there you go.
Got it. So you're just saying it's the most optimal tool for now, and that's what we're
Absolutely.
using. Yeah.
I mean,
Got it.
absolutely. And again, to me, it's math. I also, honestly, Jared, I wonder how many people who are, so basically hysterical about AI or whatever now know what matrix is, if they know basics of linear algebra.
Neural networks. Yeah.
Yes, or they just repeat AI several
Yeah.
times per sentence.
Yeah.
Because
Yeah.
but really what makes things going is actually mathematical developments. So
Yeah. No, for sure. I mean, yeah, but I mean, what's sexy to sell, right? Like saying neural networks and computer vision, or is it to say AI? And then what's sexy is usually ends up selling. So I guess that's why I've been using the term AI. We see in the news, every time I open news, I'm like, oh, AI, application of AI in radiology, or application of AI in like detecting eye disease or whatever. And there's always this something new. Or like recently, there was a startup that's developing a new tool for tooth decay with AI to predict tooth decay before it happens. So I feel like AI is just everywhere, and I feel like diagnostics is really like trying to, and which also kind of makes sense because diagnostics essentially you use so much data, and if there's a lot of data, that's where you can use the models, right? Yeah, it's been a wonderful conversation. I genuinely really enjoyed it. Do you have anything else to say to our viewers or about your company startup?
no, thank you so much, Jared. I'm really grateful for you to this opportunity. I'm actually very happy to. That was definitely the nerdiest conversation I had in a few months. So that's great, because usually we really are not talking into any of these details.
Yeah.
But yes, so that was great, and I hope that everyone, keeps optimism and is being, technological enthusiast, because we can all make this planet and this life better. We just should pick the right goals and keep going to them. We can do it.
Great. Thanks, Jenna.